Hello students, today we will see Histology of Muscle Tissue Part 2. In Part 1, we have already seen the basic features of muscle tissue and we studied the histological features of the skeletal muscle. Now we will see cardiac muscle. Cardiac muscle shows striations, is involuntary in action. The cardiac muscle fibers are located in the heart and they show autorhythmicity. That means that they contract on their own without any stimulation. They are supplied by autonomic nerves. Sympathetic supply stimulates the muscle fibers while parasympathetic supply inhibits its activity. The myocytes are connected to each other by densely staining cross bands called intercalated discs, and these are specialized cell junctions. The cardiac muscle fibers are short, thick and branching muscle fibers. Each myocyte measures 50 to 100 micrometers in length and 15 micrometers in thickness, has a single centrally placed large oval nucleus. Myocytes are joined to each other by the intercalated discs. The muscle fibers show alternate dark and light bands, that is, they do show cross striations. However, these are less prominent as compared to those that are seen in the skeletal muscle. Cardiac muscle shows less number of myofibrils as compared to the skeletal muscle, and they accommodate more amount of sarcoplasm and mitochondria. The sites where we see cardiac muscle in the body: heart tissue, superior and inferior vena cava and pulmonary veins. This here we see a low power field view of the cardiac muscle. We can see the muscle fibers and we can also appreciate the branching pattern. Another view seeing the cardiac muscle fibers, and we also appreciate here that these are short, thick and branching fibers. Another view showing us the branching pattern, and we can also see the large oval single nucleus which is present in each myocyte. The salient features for spotting here are presence of long cylindrical muscle fibers, presence of centrally placed oval nucleus and presence of intercalated discs. We then go on to study the smooth muscle. These are non-striated and involuntary in nature. They are supplied by autonomic nerves, that is sympathetic and parasympathetic nerve supply. The muscle fibers consist of elongated, spindle-shaped muscle cells with tapering ends. Cytoplasm stains eosinophilic due to presence of myofilaments. Nucleus is elongated and centrally placed. The cells are so arranged that they are running parallel to each other and are placed close to each other in the form of bundles. And these bundles may be longitudinally or concentrically oriented to the long axis of the organ. In the bundles, the myocytes are arranged tightly so that the thick central part of the cell lies adjacent to the tapering ends of the other cells. Thus, there is close packing of the myocytes to give rise to a sheet or a bundle of muscle fibers. Sites where we see smooth muscle in the body: wall of gastrointestinal tract, wall of blood vessels, wall of respiratory tract, urinary tract and the erector pili muscle. This here is a low power view showing us the smooth muscle fibers. We can see that there is hardly any space between adjacent myocytes, so there is close packing of the muscle cells, and we can see an elongated nucleus in the myocyte. The salient features for spotting here would be presence of long spindle-shaped myocytes in the bundles presence of central elongated nucleus, and absence of striations. Let us now see the differences between the three types of muscles that we have studied: skeletal muscle, cardiac muscle, and smooth muscle. Now, the muscle fiber in skeletal muscle is long, cylindrical, and unbranched. In cardiac muscle, the fiber is short, cylindrical, and branched. And in smooth muscle, the fibers are elongated, spindle-shaped, and unbranched. Nucleus. Skeletal muscle is multinucleated, nuclei being flat and peripherally placed. Cardiac muscle fiber has a single oval centrally placed nucleus. Smooth muscle fiber has a single elongated centrally placed nucleus. Striations are well-defined cross striations in skeletal muscle. These are poorly defined cross striations in cardiac muscle, and striations are absent in smooth muscle fibers. Cell junctions are absent in skeletal muscle. They are seen as intercalated discs in the cardiac muscle, and they are seen as gap junctions in the smooth muscles. Innervation of skeletal muscle is done by motor nerves, while for cardiac muscle and smooth muscle it is done by autonomic nerves. Locations where we see skeletal muscle in the body, these are seen as muscles attached to the skeleton, so skeletal muscles, tongue, esophagus, and diaphragm. Cardiac muscle fibers are seen in the heart, superior and inferior vena cava, and the pulmonary veins, while smooth muscle fibers are seen in the vessels, tubular viscera, and organs. This year we see the histological features of all the three types of muscle fibers. This here is a skeletal muscle fiber, elongated, long fibers with multiple nuclei placed at the periphery and presence of cross striations, and these fibers are unbranched. This is cardiac muscle fibers, where we can see these are short, thick, branched fibers having a single oval nucleus centrally placed. And this here we see close packed muscle fibers seen in the smooth muscle, each fiber being elongated, spindle shaped, having an elongated single nucleus which is placed centrally. And we also see here that the central area of one myocyte lies adjacent to the tapering ends of the other cells, giving rise to the close packed structure. Thus, we have studied the three types of muscle fibers. For the exam, the slides which we have is the longitudinal section of skeletal muscle, longitudinal section of cardiac muscle. These are the two important slides for the exams. And for further reference, we can read Sontake Yogesh, Textbook of Human Histology by CBS Publishers and Distributors Private Limited, New Delhi, 2020, page number 92 to 105. Thank you.